This protocol provides a means for culturing bacterial cells at the single cell level inside giant vesicles. It's easy to develop the giant vesicles, and only a very small volume of sample solution is required to prepare the vesicles. Demonstrating the procedure will be Yuri, a PhD candidate from our laboratory.
Begin by adding 20 microliters of freshly prepared POPC solution and 4 microliters of freshly prepared biotin PEG DSPE solution to a glass vial. Evaporate the organic solvent by airflow until a lipid film is formed. Place the film in a desiccator for 1 hour to completely evaporate the organic solvent, and add 200 microliters of mineral oil to the vial. Cover the opening of the vial with plastic paraffin film and sonicate the vial contents in an ultrasonic bath at 120 watts for at least 1 hour. For small vesicle preparation, create a lipid film as just demonstrated and add 200 microliters of 200 millimolar glucose in LB medium to the film. Then sonicate the film at 120 watts for at least an hour, followed by extrusion of the resulting giant vesicles into small vesicles using a mini extruder and a polycarbonate membrane with a 100 nanometer pore size. For bacterial cell preculture,